Hi everyone, welcome to Module 3 Labs. I'm going to be walking you through the different experiments that are in this lab so that way you kind of get a general feel of how you should be doing them. So the first experiment is about Punnett squares. Now you should have already reviewed the videos and the textbook over how to complete Punnett squares before you begin this lab, so that way you understand how to perform these genetic crosses. So I have my instructions printed out here, and then you're also going to need to have your combined lab report open. Now I have a paper copy just because it's for me to show you. Yours will be online. So have your tablet or your computer open while you're doing this. You don't have to worry about spills or anything like that, so your electronics are not in danger. So starting with the first experiment, when you look at the procedure, it says part 1 Punnett squares. It correlates to the part of your combined lab report that says part 1 post-lab questions. So the first procedure says set up and complete Punnett squares for the two crosses. You have two capital Y's, which is homozygous dominant, and then you have a heterozygous parent, which is capital Y, lowercase y. And you're going to go ahead and perform that cross. So what you should end up seeing is put one parent on top and then one parent on the side, and then the squares inside are the offspring, or the potential offspring. So what you should end up with is, in this particular cross, you're going to have 2 out of the 4 chances of having a homozygous dominant offspring, and then you have 2 out of 4 chances of having a heterozygous offspring. But 100% of them are going to be the phenotype yellow because they've all inherited that dominant letter. And so you just go ahead and perform the 2 crosses, and then you answer the question under the post-lab questions underneath those crosses that you perform. So you don't really need any materials yet. Now the materials that you need to get ready for the next part is these beads. So you have green, blue, red, and yellow. Now some lab kits in the past have come with orange beads instead of yellow beads, or orange beads instead of green beads, and that's okay. You just use what you have, but hopefully they sent you the right ones. And then you need 2 beakers, one beaker labeled 1 with the dry erase marker and 1 beaker labeled 2. So we're going to go ahead and go to part 2 of the procedure. And in part 2, it tells you to label these 2 beakers, and then you're going to put 50 blue beads and 50 yellow beads into beaker 1, and 50 red beads and 50 green beads into beaker 2. So I'm going to go ahead and do that. Okay, so I have mixed up my beads, and I currently have beaker 1, which has yellow and blue beads, and then I have beaker 2, which has green and red. We're going to set beaker 2 aside for right now and just use beaker 1. We're just going to mix them up a little bit. So, whoops. Escaping gene, just so that they're, kind of mixed in together. So the part that you're on in your combined lab report is this part 2, where you just have beaker 1, okay? So 50 yellow beads and 50 blue beads. Now, you need to remember that yellow is dominant to blue, okay? So if you pull out a yellow bead, it's dominant to blue. And what you will do, according to your instructions, is without looking, you're going to pull just 2 beads out. So I'm not going to look, and I'm just going to randomly grab 2 beads. So I got 1 yellow and 1 blue. So the genotype of individual 1 is capital Y and lowercase y, sorry. So I put that into my table. And then I'm going to just set them aside, and then I'm going to randomly pull out 2 for individual 2. And I got another yellow and blue. So capital Y lowercase y. Okay, so that's parent generation 1 on your combined lab report. Parent, now this is going to be different for you because you may pull out, you may pull out 2 yellows or 2 blues. So everybody is going to have different results on this. So this is just my random results. So here I'm going to do parent 1. I'm going to do it one more time. 2, so I got 2 yellows. So that's capital Y, capital Y. And then I'm going to do genotype of individual 2 for the parent 1 generation. And I got another 2 yellows. Okay, so 2 capital Y's. So I'm going to go through and I'm going to complete this chart with the gametes that I pull out. And then I'm going to fill in table 2, which asks you, hey, what's the possible genotype, offspring genotypes? What's the possible phenotypes? Now, in order for you to get those answers, you're gonna have to perform a Punnett square for each generation. So I'm gonna have to do a Punnett square for that first generation that I got, the 2 heterozygous. So I'm just gonna do 1 to give you the example. So here's my Punnett square. And remember, the first parent generation, I got heterozygous. So I'm gonna go ahead and put my 2 parent genotypes there. And I don't have to see your Punnett squares. If you want to show them to me, that's fine. But I don't have to see all that work. I trust you to do what you're supposed to do. And so the possible genotypes that I have in that parent 1 generation is I actually have 2, and the Y's are kind of difficult because you really have to mark the lowercase ones really well. So I have a homozygous dominant possible, I have 2 heterozygous genotypes possible, and 1 homozygous recessive possible. So the potential phenotypes that I have is both yellow and blue, because I could get that recessive offspring. And then the ratio that I got for that is it's a 1 homozygous dominant ratio to 2 heterozygous, because there's 2 out of the 4, to 1 ratio of the homozygous recessive. And then the phenotype ratio is you have 3 out of 4, so you have 3 out of 4 yellow and 1 out of the 4 blue. And that's all based on that, just on using that first parent generation and doing that 1 Punnett square, okay? And then there's all my answers for that parent generation. Now you're gonna do the rest, and remember, each one of these has to have a Punnett square in order for you to fill in the next part, okay? So you're gonna randomly draw for this next part, and then do a Punnett square, and then fill in your information. So that's how you complete this part of the experiment. And then you have some post-lab questions that you'll have to fill in. Hey everyone, so now we're on the dihybrid cross part of this experiment. So in the first one, we were just looking at yellow and blue beads for 1 trait, which was color. The offspring could either be yellow or they could be blue. Dihybrid is a little more complicated because now you're looking at 2 traits at one time. So we're gonna bring in our red and green beads to represent the shape of the kernel. So it's either gonna be smooth or it's going to be wrinkled. So if you pull a green bead, it's smooth, and if you pull a red bead, it is wrinkled. So in your directions, under part 3, dihybrid cross, you're going to, without looking, pull 2 beads from each beaker. So it's similar to what you did before. So without looking, I pulled 2 beads and I got 2 blues. Like I said, your results could be different, and that's totally fine because it's just random chance. So I'm going to go ahead and put little y, and genotype of individual 1. And then I'm going to randomly pull 2 beads out of here. And I got a red and a green. Now, green is dominant to red. So I'm going to have capital S for the green and a lowercase s for the red. And I put it, and this is all genotype of individual 1. Okay, and I'm just going to set them to the side. And now I'm going to do the genotype of individual 2. So I randomly pulled 2 beads. I got 2 yellows. So capital Y, because that's the dominant color. And then I'm going to pull 2 out of here. And I got another heterozygous. So I have a green and a red. So I have capital S, lowercase s. Okay, so very similar to what you did last time in the monohybrid crosses, you're going to have to perform those Punnett squares in order to get your potential offspring genotypes, phenotypes, and the ratios. So I will have to perform a dihybrid Punnett square for this parent generation. Now the dihybrid has 16 boxes. Okay, so it's a little bit more complicated, so make sure you review the videos that I posted about how to perform a dihybrid cross. And so you'll do a Punnett square for each one of these generations. And for each one, you're just gonna pull out, 2 separate beads for each one and write down what genotypes you pulled, okay? And it'll all be different. So hopefully that helps you kind of cut down on any confusion that you might have about what to do with these beads. Hi everyone, so we're on the coding experiment now for module 3, and you're going to be using these beads. Unfortunately, we don't have real genes to use for these experiments, so that's kind of what these beads are for. They're to help model what you're going to be doing. So in your post-lab experiment coding, it tells you that you need to use these beads to come up with a code. So for each letter. So I just did random codes with using the beads. So like for letter C, the code, it's a triplet code, because in real life, in your DNA, your coding comes in a codon, which is 3 letters. So for like letter C, I just said, oh, letter C is represented by a red bead and 2 blues. Letter E is represented by a red bead and a green and a blue. You don't want to repeat anything, so you want to make sure your code, doesn't, like, there's not another red, blue code anywhere. And you also want to make sure that you do a code for start and stop and a space. And in your procedure, so after you record your code, you're going to be using a sentence called "The mouse likes most cheese." So you're going to spell that out with your beads. So we're going to start with the T, which I put as a red, yellow, because remember, each letter is represented by 3 beads. So that's my T in the mouse likes most cheese. And then my H, I had a blue, green. So there's my TH. And then my E, I had a red, green, and a blue. Okay, so there is the word "the" in that sentence, "the mouse likes most cheese." Now you're going to need to put a space. Oh, you know what? I forgot to put my start. Whoopsie. So I need to put my start there, because we're starting the code. So you can see you'll end up with a lot of beads. So you're gonna need some space when you do this. Okay. All right. So There's the start. You don't need to put a space for these. So start, T-H-E. Now I need a space, the beads that represent my space, and then I'm going to spell out mouse, and then a space, and then likes, and then a space, and then most, and then a space, and then cheese, and then stop. Okay? Once you have that done, you're going to count out how many beads you used total and answer that in the lab questions. And then it's going to have you start removing beads and seeing what happens when, and that's going to represent mutations. And once you start removing the beads, it'll most likely change your sentence. And sometimes the sentence will still make sense and sometimes it won't. But the important thing to kind of focus on here is, what do the mutations do? And which one is the most harmful? Because your DNA is being mutated all the time by environmental factors, by lifestyle factors, and it's important to remember that there's things in place to help stop those mutations or to repair them in your DNA. Hey everyone, so now we're on experiment 2 for module 3, which is transcription and translation. You still need your beads, and you need these lovely poppet beads for your next experiment. And what the smaller beads are representing is the nucleotides. So like the A, the T, the G, the C in your DNA. And the poppet beads are amino acids. So remember that your nucleotides come together to make amino acids, and then amino acids come together to make proteins, which is what you need in order to survive. So they want you to come up with a 5-word sentence using no more than 8 different letters. So my 2 daughters just got in trouble for fighting with each other. So the sentence that I came up with is, I am mad at you. And that's, 5 words and no more than 8 letters. So same thing like you did in the coding experiment, you're going to have to come up with a code for each one of your letters, a 3 code for each letter, and then kind of start just putting them out, just like you did with the mouse likes most cheese in the last lab. So I'm going to go ahead and start with my start. So I'm keeping my start the same, which is just 3 reds. And then my I is, I put blue, green, yellow. And then I need a space because, right, there's a space between I am. So my space is just 3 yellows. Okay, so here's my space. So here's my I, my space, and now we're going to spell out the word am. So am is red, green, blue. And I just made this up. Okay, so you make up your own code. You don't have to, there's no like special bead code. The only thing is make sure you're not repeating. So that's A, and then M is green, red, blue. Okay, so this is just to kind of get you started. So here's my start of the code, my I, my space, and then AM. So I am, and then eventually I'm gonna, put my space and I am mad at you, right, for my whole sentence. And you're gonna count how many beads you used, and then it says to assign a poppet bead to represent each codon. You don't have to assign a poppet bead for the start or space or stop. So each one of these is gonna get a poppet bead. And the color really doesn't matter. You can put different colors for different different nucleotide codes. So for instance, I is red, my A is pink, my M is black. Okay. And what you will end up doing once you complete your sentence is you'll connect these, and you'll have a chain of amino acids. So this is one amino acid, the second amino acid, and the third. And eventually what these do is they build together to make a protein. And that's pretty much it.